Okay, today's topic is the sensory system. This springboards off of the nervous system. As you remember, all of the nervous system stuff we did was motor. And I said, we started with the brain and we went through well, first we started with tissues. What was nervous tissue? We learned about a neuron, neuroglial cells, what their function was, yada, yada, yada. Then we did the brain. We took the parts of the brain. We talked about how the brain functions. We talked about gray matter. We talked about white matter. And then we went to cranial nerves, which are part of the peripheral nervous system out of the brain. Those are both sensory and motor. And then we did the spinal cord. We did the anatomy of the spinal cord. We talked about cauda equina, filum terminal. We talked about the meninges, CSF, all that kind of stuff. And then we talked about the spinal nerves. We talked about plexi. And then we did, that was all motor. And then the very end, which you just had videoed online, was the autonomic nervous system. That's the one where it had me up in the corner and the PowerPoint. I was going over what I wanted you to know on the PowerPoint. The autonomic nervous system, remember, comes out of the lateral horns of the spinal cord, still exits, because it's motor, it still exits out the anterior horn through the rootlets, through the anterior root to the trunk. From the trunk, it either splits out the back, and it's called the posterior rami, or it comes out the root, not the very front. The very front is autonomic sympathetic. And then we had the sympathetic chain of ganglia. I went over all of that. Okay, all motor. Sensory, we saved for last. It's straightforward. It's just like it. Still has neurotransmitters, still has neuroreceptors. The only thing is, instead of going out from the spinal cord, it's coming in from the spinal cord and then going up to the brain to be processed. That's it. There's still myelinated axons. There's still nodes of Ranvier. There's still saltatory conduction, still -70, -55, +30, all those things still in place. So there's no sense in going over it again. So I'm pushing that to the side, say, it's exactly like motor, just make it sensory. We're good to go. Okay? You know that you have sensory, autonomic or visceral, and somatic. Exactly the same. So what we're going to do with this block of time is we're going to go over the senses, the receptors, the receptor of taste, of smell, of vision, of hearing. And obviously the labs that correspond with that are the eye and the ear. And I'll go over those for you. Okay, so here we go. Now it says up here, that I'm just going to be almost to the edge of the vision. So you'll just see my arms. We're going to talk about the senses. The sense of vision is conducted by what? Here's what the body has done. Instead of all parts of your body being able to see, it says we're going to establish one thing to be your vision, and we know it to be your eye. If you're having difficulty seeing these words, they're a little bit blurry, go into the program and look under HD. HD. That should clear it up. Okay, back to this. Sense of taste. Where do we get our sense of taste? Mainly the tongue. Okay, now there is some taste buds up in the roof of your mouth, roof of your mouth, on the palate. Sense of touch. Okay, now we talked about touch when we did skin. Right at the surface, below the epidermis, what did we have? Meissners corpuscles, remember that? And I've got that over here. Meissners corpuscles. We also went very, very deep, and we had another one, Pacinian. Pacinian corpuscle, also called lamellated, is for pressure. So it's deep. It's all the way down by the subcutaneous layer. And that was Pacinian. And then I told you, remember on the model of skin, there was some other red structures in there, and I said, don't worry about those, but those are Ruffini and Merkels. Well, now I'm going to tell you about those. Okay, but anyway, all of those deal with what we call the sense of touch, whether it's light touch, heavy touch, pressure, three-dimensional structure, stretching. Ruffini is for stretch. When you stretch your skin, it's going to send an impulse up to your brain that's a learned behavior of skin stretching or contracting or something along that line. Merkels is for three-dimensional shape. So if I put this in my hand, hold it out to the side, move it around, I can feel a tube shape with some structures on it. That's Merkels and Ruffini working together, stretching and three-dimensional. Do you understand? There are others, but these four are the ones I want you to know for touch. Okay? And then we have smell. Our sense of smell is through our nose. Okay? Now I know you're thinking, well, when I eat something, I can taste it, but I can also smell it with my the taste. It's a combination. Remember, your nasopharynx is connected. We'll get to that. The sense of hearing, we know, is the ear. And the sense of balance, if you remember with the brain, balance was the cerebellum. We talked about it, fine motor control, or we talked about the cat part of the brain. That's not the kind of balance we're talking about. So what I usually put for balance is I put head orientation. Head orientation. So when we move our head to the side, we go on. I know my head's to the side. If it's attached to your body, you're moving. You see where I'm going with this. Okay? That comes from your inner ear. And we'll do that one. So balance and hearing are ear. Okay? Now we have receptors. Remember, receptors are dendrites, neurons. But the dendrite portion of the neuron is the part that where it begins. We have neurons, dendrites, that are sensitive to photo, which is light. And we call those photo either ceptors or photoreceptors. So that goes down here. Okay? So the photoceptors or photoreceptors are placed in the eye. Chemoreceptors, chemicals. Okay? And it's chemoceptors or chemoreceptors. It's going to be that for all of these. And they depolarize, remember that? They depolarize when a certain chemical stimulates them. Mechanoreceptors, eardrum, touch, mechanoceptors or mechanoreceptors. Noci, pain receptors, like the females do to us. They hurt us. Pain. Thermoreceptors, temperature. Baroreceptors, pressure. And proprioreceptor is appendage orientation. We call it body orientation, but that's knowing where your body's at, your appendages, your fingers, your feet, your legs, your arms. You may have heard of phantom pain, where an amputee has an appendage removed and they can still feel it. I'll talk about that. I'll do it right now. Knowing where our appendages are is a learned response. So when we're born, we're constantly looking at our fingers, we're moving our fingers, we're touching, we're hitting ourselves, and that's putting in information into the brain, storing as either occipital lobe with vision or sensory with the parietal lobes and so on and so forth. When we feel our feet, our brain knows what to think. It knows how to feel our feet and separate our feet from our knee, from our leg and our calf and so on and so forth. Once that's removed, your brain still thinks it's there. So if you have a pain coming from down there meaning lower part of your body, your brain hasn't learned by looking down and seeing that it's not there, that it's not coming from your foot. Because you know the nerves are being fed into a bigger tube of nerve, sensory nerve coming up. So you have to start off with little ones and they're fed into this big one and it's confused. Okay, so when you say, oh, my foot hurts, you look down, you don't have a foot, it's a clue that may not be it, and you do that and pretty soon that goes away, and that's called phantom pain. Proprioception is, remember I had you take your fingers, 2 fingers, and put it behind your head? I said, put your hand behind your head, point 2 fingers, bring it forward, did you get it right? And you did, and that's because you have proprioceptors in your tendons, in your muscles, and your joints, so you know what's going on with your distance, your appendages out at a distance. You notice when you walk at night, when you can't see the ground, you know where your foot's at, you know where the floor is coming because of proprioception, orientation of body or body orientation. That's what that is, okay? Body orientation. Okay, first sense we're going to go through. First sense we're going to go through is the sense of taste. Proper name for taste, gustation. Now we're not going to use the palate bone or our palate or our palatine bone. We're going to just say the tongue. Okay? Now, the things on your tongue, when you stick out your tongue, those little rises you see there, little rises, okay? You probably think, oh yeah, those are my taste buds. No, they're not. Okay? Here's your tongue from the side. Here's your tongue from the top view. If I take a section of this out and draw it, you're going to find out it goes like this. Okay? These are not taste buds, these are papillae. Okay? Papillae. Now, if I take a section of papillae out, I'm going to change color on you. It's going to look like this. Got it? These are taste buds. Anytime you see the word taste, you can substitute gustation in there. So those are gustatory buds. Okay? Now I'm going to take one of those gustatory buds and I'm going to enlarge it. Make it nice and big for you. Okay? This is a gustatory bud. Okay? Taste bud. This is a gustatory or taste pore. Now, this being tissue, we know it's made out of cells. I'm drawing in the cells right now. How many of you are in your pajamas right now? You better not be drinking cocktails and eating stuff, gluten. Make me feel bad. I wore my pink shirt for you. Okay, get back serious. These are cells. Not all of the cells are attached to a neuron. And we call these gustatory cells. Okay, this was a gustatory bud. This was a gustatory pore. The blue ones are gustatory cells. The green ones are just called support cells. Got it? Now the gustatory cells, remember it's a dendrite, cell body, axons are all connected together. And this forms cranial nerve. And with the sense of taste, there are two of them. Cranial nerves number 7 and number 9. 7 is facial, 9 is glossopharyngeal. 7 is facial, glossopharyngeal is number 9. That's the two that I just drew up there for you, cranial nerves. And you saw where they come from. Okay? You have an axon, an axon, an axon, an axon, pretty soon you got a nerve. And it's not a tract because it's a cranial nerve, part of the peripheral nervous system. Are we good? Remember, on your exam, 3 things with the cranial nerves. 1 is its Roman numeral. 2, its name. 3, its common function. Okay? So 7 and 9's common function is sense of taste. Good? All right. Let's do some checking off. We know what papillae are. That's the orange up there. We know what a taste bud or gustatory bud is. It makes up the papilla. Taste pore is the hole in the middle. Now we're going to do some mechanics. Understand that fluid is coming across here in the form of saliva. You've eaten something. You've been chewing it. It's been going across your tongue, and that's those red lines right there. Okay? In order to get a taste, you got to get the fluid down in here. So we add saliva to it from the 3 salivary glands. And to help it come down, we put a rod up there. That rod is made out of a protein, and it's called a gustatory or taste hair. So once this strikes it, and I'll do it in a different color for you, once this saliva chemical combination strikes the hair, it enters the taste bud. Sticking up here, I'll do it in black. Are dendrites. Now, these dendrites don't just depolarize with chemical, it's specific chemicals. Okay? And you're going to find out they depolarize with, we have 5 of those chemicals. Okay? If the fluid that comes in here is glucose, and this only depolarizes with a bitter taste, even though the fluid's in there, nothing happens. Nothing comes down the cranial nerve. If this is sucrose or glucose in here, and this is sensitive to it, then this will say you have, that saliva has glucose in it, and you'll have a sweet taste. Do you understand? Okay. So that was taste pore, taste hair, gustatory cells, support cells. We good? And then the cranial nerves that are involved. Now let's go back up to something you're probably familiar with. The sense of taste. What senses do we have as humans in our mouth? Okay, at the end of your tongue, this is the end of your tongue, this is sweet. Overlapping it on the forward anterior margin is salt. Back on the margins on the base, it overlaps a little bit, is sour. And then across the back, is bitter. And you learned the 4 senses, right? Sweet, salty, sour, bitter. I'm going to have a simple question on the exam on it just to make sure that you know where it's at on the tongue. Then there's another one you may or may not have heard of called umami or umami. And that sense of taste is savory. And you may have heard of savory when they talk about meat, especially beef. Oh, it has a savory taste. Okay? And that depolarizes with the sense, with an amino acid. And the amino acid is glutamic acid. So when you eat a protein that has glutamic acid in it, you're going to taste it. And what makes it so robust in your mouth is it's all over the tongue. It's not just located in the margins or front or to the side or something along that line. It's all over. So the savory taste, which is umami or umami, is all over the tongue. Okay? Glutamic acid is the amino acid. You may have heard of it, not even known it. MSG, monosodium glutamate, glutamic acid. So the stuff we hear a lot of times associated with Chinese food, okay? You can buy it. Just like you buy salt, pepper, sugar, it's in the same section. Sometimes it might even be called accent, A-C-C-E-N-T. It's a yellow, a white and red, little like a salt shaker, about this big around, about that tall. Okay? I was in Smart and Final. I found a big jug of it. So I took it home and I put it on something. I don't remember what it was. Might have been on chicken or something. And the wife says, wow, this really has a lot of flavor. I grinned. Okay? And then finally, of course, I had to tell her that I put MSG on it. Most people freak out over this. Do you have salt in your diet? Do foods have salt on it? Yes. This happens to be monosodium and glutamate, which is an amino acid, which is in proteins. So is this going to hurt you? No. But didn't you hear about, oh, this has MSG, it's not good for me. It's like if too much of anything's not good for you. Yeah? Okay. All right. Now, I'm going to pause it. I'm going to erase this to give me some space so I can do the sense of smell. Okay, we're back. Now we're going to do the second of the four senses that we're going to cover, the sense of smell. I take you back to the sense of taste, it's exactly the same way, only we put it up in your nose. Instead of having the 5 sub-senses, in other words, the sense of sour, sweet, bitter, that kind of stuff, we have a lot of them. It's still a chemoreceptor. It's still a chemoreceptor. So what I put up here is the sense of smell is properly called sense of olfaction. And I also put these two terms on here, cilia and columnar epithelium, ciliated. Didn't want you to forget. Remember the trachea? Pseudostratified columnar epithelium that was ciliated, so we put cilia out on the front of it. Ciliated, pseudostratified columnar epithelium. And an occasional goblet cell in there. Remember that? For you allergy sufferers, you know what goblet cells are doing to you right now. Okay, so that's the reason those are up there. Now you'll see a little bit olfactory neurons, bipolar, cribiform plate, synapse. I'm going to draw that. Okay, got to concentrate here. Here's a nose. This is where I get messed up, is on the lip. Here's the eye. And there's the forehead. Here's the eyeball. It's looking pretty good. Okay, here's the lips, mouth, lip, chin, neck, one tooth, Willie Nelson concert. Nares, respiratory. You know what nares are? Okay, not too bad. Here, we'll give 2 teeth. Make him a young Willie Nelson concert. Now, we have, with the sense of taste, we were tasting a liquid or solution. We're tasting chemicals in a solution. With the nose, we're taking the same chemicals and we're putting them into a gas. So I'll put the gas out here like this. And the gas comes up your nose along with the air. Okay? Right here, now I'll change colors. Right here is the floor of the skull. The floor of the skull. So up here is where your brain is at. Down here is where your nasopharynx goes down. Remember? The nasopharynx, oropharynx, laryngeal. We have the larynx here. Hope this looks familiar. Okay, and we had the esophagus going back off of that. And these were the cartilage rings. This is the trachea. Okay, if that doesn't look familiar at all, I don't know what to tell you, man. Okay, McDonald's is hiring. Now, so we had spoken a little bit about taste. Sometimes if your nose is plugged up, you can't smell very well and stuff. That's because it's connected. Anyway, this is the floor of the skull. So in lab, if we take the skull off and look down on the top of it, we'll see right toward the front a little plate that looks like this. And there's a raised portion in here. This raised portion is called crista galli. And the side of it, I'll do it in black so you can see it better. There's holes. And some of you have either rented or bought your own skeleton. Right now would be a good time. Pop that skull cap off, look down, you'll see what I'm talking about. This is called the Cribiform Plate. Damn. And here's where I'm at. Okay? The crista galli separates the right side and the left side. In fact, you know what? I'll even show it to you right now on the brain. You see those? This is where I'm talking about, right there. Okay? There's the left side, there's the right side. In the skull, right here, I've even got that. Maybe I don't. I'll get back with you on that one. I'll pull out a skull and show you where the crista galli is. Never mind. I'll do it right now. I'm going to pause this and go get it. Okay. I got this here pretty quick, huh? I'm going to take off the skull cap. Skull cap. This is the floor. Remember, this is the base. This is the floor. Right here is the place I'm talking about. In the center is the crista galli. That separates the right and left olfactory nerves. And right there is, you can't see it too good because this is plastic. Those are little holes. If you took out that rectangle right there, that would be the cribiform plate with the crista galli separating it. Do you see? Okay. It separates these two things. Okay. That being said, let's get back over here. So I'm going to draw the crista galli from the side. I drew it from the top. Now I'm going to draw it from the side, and there it is. And there's the holes in it. What happens is that on the brain I showed you, there's a bulb called the olfactory bulb. There it is, olfactory bulb. And there's tissue here. And made up of cells. Doesn't this look just like a taste bud? And some of those cells are olfactory cells that have cilia on them that detect the sense of smell. And they extend down through the cribiform plate. So when the odor comes in and starts its way down the pharynx, down to the trachea, whatever chemicals are there, I'll do it in red so it's dramatic, will touch the dendrites, the sensory neurons, and then send that impulse as learned into here. That goes into being processed in the temporal lobe. Remember that? The sense of smell. Now let's look over here at the checkoff list. Olfactory neurons. Remember there's 3 types of neurons, unipolar, bipolar, and multipolar. These happen to be bipolar. Bipolar. So if I drew this, it would look like this. Okay, there is the cell body. It's in that bulb. These are the little blue lines I drew, and this forms the olfactory tract, olfactory nerve number 1, cranial nerve number 1. And we do that as Roman numeral 1. Good? We have a whole bunch of different types of olfactory neurons that are sensitive to different chemicals. In the tongue, there was 5. Here, there's thousands. Are we good? Once it depolarizes, it sends it up as learned behavior or a learned response. So for an example, the first time you smelled cinnamon, you smelled an odor, but you didn't know it was cinnamon until you learned that that smell is cinnamon. You tie the 2 together, just like the sense of taste, just like the sense of touch we talked about. Okay? I'm going to pause it for a second, erase the board. The next thing you're going to see up is the eye. Okay, we're back. The next sense we're going to do is vision. So it obviously involves the eye. Now I have these portions right here is just some terminology I want to bring up. Let's go. Let's make this the eye socket. Sphenoid bone, maxillary bone, temporal bone, so on and so forth. This the eyeball. Coming off the back of the eyeball, optic nerve. Front of the eye, eyelash, eyelash. The eyeball just kind of sets in here. That means stuff can get in here and ultimately get back in here. So we protect it by putting a tissue there. That tissue is called conjunctiva. Here's where I'm at. Conjunctiva. Since the eyelashes are made of hair, you know the hair is a shaft and a root. The root is in a follicle. The follicle is fed by sebaceous glands that it can get plugged up and form a pimple. The pimple in an eye, pimple in the eyelash is called a stye. Here's where I'm at. Eyelash, stye. Eyelids do 2 things. Eyelids, when you close them, cover the eye to keep it from drying out. Shield the eye when dust and particles are coming at the eye and it's sensed. And also when it squeezes on the lacrimal gland, which is about right here. The lacrimal gland is your tear gland. Okay? And as it does, it's like a sponge. It just secretes out a fluid that covers the eye. And then right here in the corner of your eye is the lacrimal duct that takes it away. So when it's squeezed, it's picked up and taken away. Squeezed, taken up, picked away. This is the lacrimal gland. So it'd be sitting in my eye like this, in my head. Okay, this being the maxilla, this being part of the sphenoid bone. This is the lacrimal canal. I'm sorry, the lacrimal gland, your tear gland. Down here in the corner, which was asked on the last exam, was the lacrimal canal. Squeezes from here, covers the eye, goes into here. The eyelid is what triggers the pressure. We got it. Okay, let's check off. Eyelid, we said roughly there's two functions: to protect the eye and tears. If you want to see how to spell lacrimal, it's way over here. Lacrimal canal and lacrimal gland. Eyelash, what is the function of the eyelashes? Besides a place for the ladies to put on fake eyelashes. Or what's the other thing they do? Extensions. Those eyelashes keep dirt particles out of the eye. So, you know, when it's blowing wind out or you're painting with air paint that goes, you know, airless or paint with a paint gun, you'll see that start to cake up on the eye. That's to protect those particles from reaching the actual tissue of the eye. That's your eyelash. In a plugged up sebaceous gland is called a stye. The tissue that protects the eye, protects the eye socket from getting things behind it, is called conjunctiva. If that tissue becomes infected, it's called conjunctivitis or pink eye. If I take my lower eyelid, bend it down, you can see the conjunctiva. Okay, it actually goes all the way around. Now they say that conjunctivitis is extremely contagious. I'm going to take you into micro. When we grow bacteria on a plate of agar, the agar is a mixture of seaweed, which is the actual agar, and nutrients. And we know what bacteria likes what nutrients the best. So we mix that into the agar, and then the bacteria grow. And the bacteria, because there's so much nutrition, the bacteria can grow in exponential form until you can actually see a colony. Got it? Got it? Now, because bacteria grow on warm, moist, dark environments, what happens when you close your eye? Warm, dark, moist environment. They explode. So pink eye is not any more contagious than anything else. It's just it's so concentrated, and usually young babies get it when they don't know how to communicate that their eye hurts. They're crying. As a parent, we pick them up. They wipe their eye and show you what's hurting. And that's how it's spread. Okay? One of the ways. It's not airborne or anything. Okay, so you know what conjunctivitis is, pink eye. Okay, now, since I'm limited on space, I'm going to do some erasing. We're going to talk about the layers of the eye. There are three. Only this time we don't call them layers, we're going to call them tunics. Okay, we saw tunics once before when we did blood vessels. Okay, so here we go. There are three tunics. Here's where I'm at. There's three tunics. One, fibrous. Two, vascular, also called choroid. Don't forget there's an O in there. Choroid, two names, vascular choroid. And the third one is nervous tunic, also called the retina. Okay, so I'm going to draw the fibrous tunic. There it is. And out in the very front, it kind of protrudes out. That ain't very good. Try that again. Okay. Don't look at that little divot. That don't mean anything. He just got poked in the eye. Okay. This whole thing is called the fibrous tunic. Whole thing's called the fibrous tunic. It's subdivided into two parts. The white of the eye, which is called the sclera. So I'll do it like this and like this. This is called the sclera. You know it as the white of the eye. Sclera. In the front is the clear portion called the cornea. So the fibrous tunic, which is the outsidemost tunic, has two sublayers. One, the sclera, two, the cornea. Using the same model, you can probably see this little clear part here. This is the cornea. This is the white of the eye, called the sclera. Both of them together, fibrous tunic. That's it. That's it. Next one. Vascular or choroid. Two names, same thing. Associated with that, I'm going to do it red because some of it's muscle. I'm drawing the second tunic. And you notice I kind of put a bulb on the end. Let me do it a little bit more better here. And since blue eyes are best, we're going to put blue eyes on this guy. Okay, now I'll do it in red so we're consistent. This is the vascular or choroid tunic. And what do we know about vascular? Blood vessels. Yes, this is where the blood vessels are at. Okay, now I want you to zero in on this portion right here. This is called the ciliary body. Here's where I'm at. Ciliary body. Ciliary body has two subdivisions. Ciliary muscle, ciliary process. Ciliary muscle is the blue part. Also known as the iris. Okay? The colored portion is pigmented muscle. The pigmented muscle in my eye is blue. The pigment in your muscle may be blue or green or brown. Do you understand? That is the iris of the eye. It's part of the ciliary process. Part of the ciliary body, and it's called the ciliary muscle. So under ciliary body, there's two parts. The iris, which is the muscle, and the next one, and I'm going to do it in red, is called the process. And that is this portion. Ciliary process, and here's where I'm at. And associated with the ciliary process, I'm going to do it in black. or ligaments. We call those ligaments suspensory ligaments. Suspensory ligaments. Now listen, the suspensory ligaments come off of the ciliary process. The iris is the ciliary muscle. Okay, one more time. The iris is the ciliary muscle. The process has the suspensory ligaments. The two of them together make up the ciliary body. And what's being suspended is the lens. The lens. So over here I put ciliary body, consists of two things: ciliary muscle, which is the iris, the colored part of your eye, and the ciliary process, which has the suspensory ligaments that suspends the lens. You got it? I'm going to show it to you on the model in a minute. And we've all heard of a cataract. If I ask you what a cataract is, you say, well, it's something in the eye. Prevents you from seeing. Okay, yeah, sort of. It is a fogging of your lens. In other words, your lens is made out of tissue. Tissue is made out of cells. Inside the cell, in the cytoplasm of the cell, is a clear protein. When it's subjected to ultraviolet rays from the sun, in other words, photons from the sun, strikes it, blue, when it strikes blue colored eyes, it affects the lens more than brown colored eyes. So evolutionarily, brown-eyed individuals lived in the savannas, out in the sun, and it allowed them to see their food and escape their predators because they could see and bright. They didn't have to squint like us poor blue-eyed people do. In the forest where light was limited, blue eyes then could pick up more light, but it's sensitive to ultraviolet rays. So somebody like me is probably going to get cataracts when you get older, but I'm not older yet. Now, cataracts is the darkening, it's crystallization of the lens cells, the protein in lens cells. Do you get it? One more time. Cataracts are the crystallization of the cytoplasm that contains the protein in lens cells. Okay? So when you have a cataract removed, they take away your lens and they give you a phony one. Do you understand? Okay. You know what a cataract is. You know the iris is a muscle, a pigmented muscle, and that muscle determines the size of your pupil. Your pupil is a hole. Here's your pupil, right there. It is a hole. Do you understand? That hole is determined by the ciliary muscle. The more the muscle relaxes, the bigger the hole, the more light it gathers. The more it contracts, the smaller the hole, the less light that goes in and strikes the next tunic, which is the nervous tunic. Are we good with those? Okay. I got to clean up that drawing a little bit. So bear with me. Make sure you look at it. Okay, what's nice about being online is you can stop it and go back. It's not going to go away like it's going to go in just a minute. Okay. I got a mess in there. Let me see if I can clean that up a little bit. I want the lens to stay there. I want the suspensory ligaments to stay there. Is that just about perfect? get rid of this line. Okay. Okay. Let's see what we got here. Now, the last tunic is the nervous tunic. You probably know it as the retina. I'm going to make a straight line for a vision. Let's say this is you out here. You're happy because you're in your pajamas. You're having a cup of hot chocolate. Okay, while I'm sitting up here trying to educate you. Okay. So this is you, your vision coming in. Comes in on a straight line and strikes it back here. Okay, now I did it in red because of the choroid layer. I did it red because it's muscle and blood vessels. Now, I'm going to do the nervous tunic in green. You can't have that covering the entrance. The only thing that lies in the entrance is the iris, the muscle, that portion of the cornea it's coming through, and the lens. Okay? Now, here's that nervous tunic. Okay? Nervous tunic. What do you think the cells are that make it up? Okay? Because you can't respond. 1, 2, 3, 4, 5, time's up. Neurons. Okay? So there are two types of neurons that make up the retina or nervous tunic. Those are rods that depolarize, detect light, and cones that detect color. These are neurons. Specialized receptors that either depolarize with light or depolarize with color. Do you understand? That's what that green line is made of. Okay, now, remember when we wanted to increase surface area? What we did was we put an extra tissue out like that, like a finger-like thing. We saw that with villi, didn't we? Okay, in the eye, we don't want to have a bunch of fingers coming out. We turn the finger around backwards and make a depression. And that area is called macula. Put that darn thing in the wrong place. Lutea. Sometimes you hear it saying lutea. Macula lutea. That is the depression to increase surface area. Got it? So why would you want to increase surface area? It's a no-brainer, so you can have more neurons in it. Yes? So your central focus, your most acute focus is right in the center of that, where I have that arrow. And that center of focus is called Fovea Centralis. And that is inside the macula lutea. One more time. The area is the macula lutea. Inside of it is a high concentration of rods and cones, especially cones, for your most acute vision. And most acute vision is called Fovea Centralis, which is a straight line. So that means when you look at something directly on, that's your best focus because you have the highest concentration of neurons. Okay? You can still see to the sides, but you notice the further you go away from the sides, the more definition you lose and also the more color you lose. Good? Good? So now you know what macula lutea is. It's an area containing the Fovea Centralis, which is there for your center, has the highest concentration of neurons. The two types of neurons that make up the the nervous tunic are rods and cones. Rods are for light, cones are for color. Okay? Now, I got to erase just a little bit right here. I don't want this junk up here. Now, go back to this layer, which is the choroid vascular tunic. It is made, all of this is made up of cells. Every layer is made up of cells. Your highest concentration is in the vascular choroid tunic, but the neurons also need nutrition. That means there has to be a blood supply. If there was a blood supply, there'd be capillaries. If there's capillaries, there wouldn't be a neuron. If there wouldn't be a neuron, there wouldn't be any vision. So we have to feed it from below. And it has to exit. So in other words, you have an exit place. And I'll do the blood vessels in red and blue. Okay, just a little bit, goes all the way around. Okay, so I just said the exit point is for the blood to go in and come out. If you have blood vessels there, you can't have a neuron. If you can't have a neuron, you can't depolarize. If you can't depolarize, you can't see anything. Okay, in addition, and I'll do it in green, the axons in cell bodies have to exit. So you have to sacrifice part of the eye to lack vision. And that is called the optic disc or blind spot. And it's, instead of being directly in the Fovea Centralis, it's down and offset just a little bit. Okay, and yes, you can determine it. And if we had a lab that we could use it, that lab, and I could show you how to determine where your blind spot is. But what I want you to know now and take away from it is, what is the function of the blind spot? It's where the blood vessels and the neurons leave the eyeball. Why is it called a blind spot? Because there's no place for neurons there. I mean, the dendrites of the neurons, no rods and cones. Do you understand? So over here, optic disc, blind spot. Are we good? Good. Okay. Now, for the model, I'm going to show you those things we just talked about. Okay. Sclera, cornea. Ciliary body. Suspensory ligaments. This person has brown eyes. Okay, this is the cornea. Behind it is the iris. Behind that, being suspended from the suspensory ligaments, lens, and then the retina. So directly in a straight line with the retina, in the back here, would be the Macula Lutea, the area with the Fovea Centralis. And the way the model is set up, if you look carefully, you can see two diamond-shaped structures. One's kind of brown and the other one is pink. You might not be able to see that. This that I'm touching, choroid vascular tunic. This, right at the end of my finger, that is the Macula Lutea. Inside, a depression containing a high concentration of rods and cones, specifically cones. Got it? Now, here's the other model we have a lot of, and this is the optic disc. Optic nerve comes out there. So I'm going to take this one apart for you. Choroid vascular tunic. See the optic nerve? Now, if you look real carefully here, pretty well done. You can see all the blood vessels. You can see red, blue, and yellow. Red, what they're showing you is an artery. Blue, they're showing you a vein. Yellow, they're showing you a neuron. Pretty good, huh? Okay. Let me pick up where we were. Optic disc, blind spot. Now we have some more stuff. Cavities. I'm going to draw. I can do it right here. We have two, the eye is made up of two cavities, posterior and anterior. The posterior is the inside of the nervous tunic, and it's called posterior cavity, also called vitreous. Also called vitreous. Here's where I'm at. And that's kind of jelly-like. So when you push on the eye doesn't just slosh around. And when you move your head, it doesn't just slosh around because it's a jelly-like fluid, kind of like cytosol is in the cell. Gives it more rigidity, but still pliable. Do you understand? That's cavity number one. Cavity number two is out here. Now I'm going to explain that one. And that cavity is called the anterior cavity, also called the aqueous. No, sorry. Let me back up. This is the posterior cavity. This is the anterior cavity. Also, no, I was right. Never mind. Also called the aqueous cavity. So far good. Just ignore that part. It was just, I needed some rock star. Okay. The anterior cavity is divided into two. And the two divisions aren't called subcavities. They're called chambers. Okay, I'm going to sneak back over here and see if you can see that with the camera. Almost. Okay. So we know the anterior cavity has two subcavities called chambers. The chambers are one, anterior, two, posterior. Now you know why this is important, because you're seeing posterior and anterior here applied to cavity. Now you're seeing posterior and anterior here tied to chambers. Be careful, because you know I'm going to ask you on an exam. The posterior cavity, vitreous cavity, that's it. But out here where the ciliary body and process and iris and all that stuff is, that is the anterior cavity, also called the aqueous cavity, that is subdivided into chambers, two chambers. And the chambers are the anterior and the posterior. Okay? The anterior chamber, now here's obviously your test question, goes from the cornea to the iris. The cornea, watch, I'll do it in orange. From the cornea, which is this shape right here, to the iris, got it? That's the anterior chamber of the anterior cavity. Anterior chamber of the anterior cavity. Are we good? From the lens to the iris. So from the lens to the iris, or from the iris to the lens, is the posterior chamber of the anterior cavity. Process that. You'll be okay. It's not that tough. So here's where I'm at. Anterior, aqueous, two chambers, anterior, posterior. Okay? Now the fluid in your anterior cavity is very liquidy. So that's why we call it aqueous. And if it stays around for a long time, it'll start to thicken. So what we do is we make it and then we absorb it. Just kind of like we did with cerebral spinal fluid. We make it, we absorb it. Are we good? It is made in a specific spot. And I'm going to draw it right here separately. I'm going to draw this part, the ciliary body. Okay, ciliary body containing the muscle, which is the iris, and this portion, which is the ciliary here's where I'm at. Ciliary process. And you know, coming off the ciliary process, oops, I got this on the wrong side. Comes out here like this. Coming down off of here is the suspensory ligaments suspending the lens. We're good with that. I drew that right here. I'll do it in green. There are cells right here that produce the aqueous humor. Cells that produce the aqueous humor. And these are collectively called, form a canal of Schlemm. Canal of Schlemm. Okay? And they produce the fluid here. And then it's reabsorbed. If the canal, now the cells are not called the Canal of Schlemm. That's what reabsorbs it. But it's the Schlemm cells that produce the aqueous fluid that's picked up by the Canal of Schlemm as kind of like a circle. Okay? Now, if for some reason the Canal of Schlemm becomes blocked or partially blocked, you'll produce more fluid, more aqueous humor than you can absorb. So you're going to build up pressure on the eye, on the lens, and the cornea. And that pressure results in a condition called glaucoma. Got it? So the Canal of Schlemm is for absorption of aqueous humor. When you can't absorb it fast enough, it puts pressure on the lens, really the muscle too, the iris, and the cornea, and results in a condition called glaucoma. And if you've seen somebody with glaucoma, it looks like their eyes are popping out, especially with blue eyes. It's not as noticeable in brown eyes, but blue eyes, it's really nasty. Okay, now some cleanup. Whenever you see the word opsia, it means vision. We know that the prefix an or a means what? Without vision. Anopsias are abnormal visionary problems. Myopsia is nearsightedness. Myopsia, believe it or not, that's S-I-A. And that's when the eyeball is elliptical. So if your eyeball is made like this, and here's your eyelashes, your normal vision would be right here. That's where your Macula Lutea would be, but it's not, it's way back here. So when you're close, you can see, because when you're close, your vision strikes back here. So When you have myopsia, you have an elliptical eyeball at birth and nearsightedness. Hyperopsia, I blew that, is the eyeball is flattened. Normally your Macula Lutea would be back here. It's not, it's right here. And the only way you can see is things far away striking back here because you can't tell the difference. And that is called hyperopsia, farsightedness. I'm going to ask you one of those on the exam. So elliptical eyeball is myopsia, nearsightedness. Flattened eyeball, hyperopsia, farsightedness. Do you understand? You can see things far away. Are we good? Hyperopsia, myopsia. Has nothing to do with getting older and losing your nearsightedness. You notice I don't have to wear glasses because I'm not old. But my vision is 2015. I can see really, really good. My grandfather had he had myopsia. He could see things up close. He couldn't see things far away. So he wore glasses to see, and when he wanted to read, he took his glasses off. Does that make sense? Okay, so two different things. Next term, blepharitis. Remember what a sty was? Some people are susceptible to a sty. They get styes all the time. What that is a reoccurring sty is called blepharitis. Trachoma, reoccurring conjunctivitis. Trachoma, reoccurring conjunctivitis. Okay, let's take the next word apart. Opsia, vision. A, without. Chroma, or chromat, means color. There you go. Color blindness. Achromatopsia, color blindness. And we have three colors, yes? So you don't have to have complete color blindness. You could have partial. You could not see reds or you could not see blues. Do you understand? Now, astigmatism. Stigmatism is when the cornea, instead of being nice and smooth like this, is jagged, has faceted. And then when you see things, what's coming in through your cornea is coming in at different angles. So the test for that, I'm going to turn the camera so you can see it. You've probably been looking at this during the semester. Okay, I'm going to zoom it in. That's as far as it zooms. We have a color chart. We have a distance chart. I said I was 2015. That's where I'm at. So I could read this line at 20 feet. Perfect vision is 2020. You could read this line at 20 feet. I can take and read that same line that you're reading at 20 feet. I can read it back at 25 feet. Do you understand? Or about there. Okay? If you were 2020, you would have to be 15 feet away from line number 9 to be able to see it, or I could see it at 20 feet. Does that make sense? That's probably an easier way of understanding it. Here's 2020. Here's 2015. So somebody at line 8 could not read line 9. Somebody who has 2010 vision, they could read this line, number 10 line. No, they'd be able to read number 11 line. That's 10 feet. This is 2013. This is 2010. They could read that line at 20 feet. I know what you're looking at right now, so you don't see nothing. I get it. Okay? I knew a cardiovascular surgeon who was 2200. That's all he could see. He's cutting on hearts. That makes you scary, doesn't it? Okay, astigmatism. This is an astigmatism chart. If you stare at the center of it, and there's 3 lines all the way around, at the hands of a clock, 12. There's 3. If you don't see 3, you have astigmatism. If those lines are not perfectly straight, you have astigmatism. If the lines are wavy, you have astigmatism. If the black line and the white line are not the same width, you have astigmatism. But you got to stare at the center. So the Fovea Centralis is right where my finger is, and the shape of those lines determine if you have astigmatism or not. Got it? So to answer your question, astigmatism is a imperfect cornea, okay? Which is what LASIK was all about. People that had astigmatism, it's the LASIK computer, the laser smoothed out your cornea so the astigmatism went away. Then they found out they could play games with it, cutting off more and allowing you to have, if you had myopsia, to have hyperopsia and so on and so forth. Okay, so you know what astigmatism is, an incorrect cornea. Lacrimal gland, tear gland. Lacrimal canal absorbs it. I showed you where it was. Lysozyme. Lysozyme is a generalized antimicrobial that is mixed up in your tears. So it's part of your tears. It's antimicrobial. It is an enzyme that breaks down foreign matter in your eye. If you break it down, you don't get an eye infection. If you can't break it down, it cannot break it down, it results usually in conjunctivitis. Okay, I'm going to pause it, erase this, and we're going to do the ear. Okay, third part we're going to talk about. Third part we're going to talk about is the ear. Now, remember when I started off this discussion, I talked about the ear isn't just for hearing, it's for body orientation, head orientation, balance. I don't want you to think that balance, we stand on one foot, or the cat part of the brain, which is the cerebellum, is what we're talking about. We're talking about the inner ear. Part of it is devoted to head orientation. There's going to be a fluid that moves in a tube, and that's going to touch neurons that are going to depolarize, telling the brain that has been trained, this is the orientation of the head. Got it? And the third thing is what I call the roller coaster part of the ear. When you go up or down really quick, or forward, backward real quick, you know how you get that feeling? That's the third part. Got it? So let's talk about the structure of the ear. Now I have a key for this, but the ear is divided into three parts. From my hand that way is the outer ear. Right between my fingers is the middle ear. Both of these are air filled. And the last one is the inner ear. That's where everything happens. All this is conduction getting it to go in here. Okay? This represents your eustachian tube that allows ambient air from the outside to get inside and equalize to the outside, because you got the eardrum in between there. And when it's plugged up because you have mucus down here and the ambient air can't get up and neutralize or equalize the outside from the inside, it starts to bend the tympanic membrane and causes pain. Okay? And when we dive, scuba dive, you have to be particularly careful, because every 33 feet is an atmosphere. So on land, I think it's 900 feet. In the water, it's only 33 feet. Puts a lot of pressure on there. Anyway, we'll come back to that. Okay. This is where you put when you clean your ears, this is where you put the, let me do it like this for you. Wind's starting to come up. This is where you put your swab, your Q-tip, to clean the wax out of your ears. And you're going to hear the word cerumen, C-E-R-U-M-U-N, cerumen. That's the wax buildup in there. Okay, ear. The ear is divided into three parts. Okay, the ear is divided into three parts. Outer ear, middle ear, and inner ear. Outer ear, this part of your outer ear, it's called the pinna or auricle. Pinna. or auricle. I'm going to check to make sure we're shooting this right. Good, 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 good. Auricle, pinna. Auricle, pinna. Put your sunglasses right here. Next part you see, it says meatus. This canal that I just showed you where cerumen builds up is called the external auditory canal. External auditory canal. You have an internal auditory canal that's inside the brain, inside the cranium. External auditory canal. Right here, the opening to that is called the meatus. So it would be called the external auditory meatus. I'll write that down for you. External auditory canal, and the opening of the canal is called a meatus, which is what's right there. Got it? Cerumen is earwax. You know, when you were a kid, put your finger in there and then you ate it. It was real nasty. I know right now you got that scrawl on your face and you're saying you never did it. So my question is, how do you know that? And your response is, because it tastes bad. Well, you're guilty because you wouldn't know it tastes bad if you didn't try it. You did. You did. I didn't. You did. Okay. So we have the outer ear. Pinna, auricle, external auditory canal, inside, meatus, opening, cerumen, earwax. And the cells that lined the external auditory canal are cerumen producing, wax producing to collect dirt stuff. It ends with the tympanic membrane. You see an asterisk here. The reason the asterisk is there is to show you it is the end of the external or outer ear and the beginning of the middle ear is the tympanic membrane. So the tympanic membrane goes up in this column and it also goes down in this column. Are we good? Now, you've heard of the little bones in your ear. Probably when you were in grammar school, you learned that there was three of them. The hammer, the anvil, and the stirrup. I hope this will focus. There they are. The hammer is the middle one. The anvil and the stirrup. Hammer, anvil, stirrup. Properly called malleus or malleus, incus, stapes. Okay, collectively, those are called the ossicles, the ear bones. The largest bone in the body was what? Femur. Very good. That's not a hard question. Smallest bone in the body, stapes, also called the stirrup. Smallest bone in the body. Should I put that on the exam? Okay, I'll think about it. Okay. Collectively, they're called the ossicles, and there are three of them. The one that touches the tympanic membrane. So that's going to take sound waves and turn them into mechanical waves, mechanoreceptor. It turns it into a mechanoreceptor, is the malleus, which is also called the hammer. Make sure you know both names. Articulating with the malleus or malleus is the second one, which is called the incus. And the last one, which actually attaches to the inner ear, and I'll put an asterisk because it ends the middle ear and begins the inner ear, is called the stapes or stirrup. Now remember the tube for equalization of the outer ear to the middle ear because they're both air filled. See, the inner ear is fluid filled. It's sealed off, it doesn't change. But when you go up and down in altitude, the outer ear and the middle ear must match or you start getting an earache. In order to do that, you have to have a tube that's protected from bacteria that can connect to the outside. So what we do is we put one opening of the tube in the middle ear, in the chamber where the middle ear is at, and the other one we put it into the nasopharynx. Okay, remember that? So in the nasopharynx, there's an opening. That tube is called the Eustachian tube. And that is for, this is on your exam, for equalization. That's called for equalization. The stapes attaches to a spot on the inner ear called the oval window. Let me show you what that looks like. Okay, this is the inner ear. This is called the bony labyrinth. This is the stapes. See the stirrup looking thing? That's the stapes. Behind that, underneath that, is the oval window. The oval window is the beginning of the inner ear, but they attach the stapes to it so you know what attaches to it. This is the tympanic membrane. Tympanic membrane. Touching the tympanic membrane is the hammer or the malleus. That is this thing in my hand. I know this is kind of difficult. We are able to put you in lab, you'd be able to touch it. This, hold on. This is the malleus touching the backside of the tympanic membrane. This is the outer ear on this side of the tympanic membrane. It articulates the malleus, the hammer, the malleus, articulates with the anvil. You see how that shape is? That shape is a shape of an anvil, like a blacksmith uses. You don't even know what that is. Anyway, then that has a little arm that comes out, and that arm articulates with the stapes, just like that. So you have the hammer touching the tympanic membrane. So the malleus or malleus touches the tympanic membrane. It articulates with the anvil, also called the incus, which articulates with the stapes, which is called the stirrup. Do you understand? So what happens is, and I'm going to try and do it, it's going to look kind of stupid, but mechanical waves are pushing on the oval window, and it's going to cause fluid in here to move. There you go. Okay? I'll come back to this. Now, if you go into what we have that used to be on Facebook, and it's still there, we've also uploaded that into Canvas, where you can see other people showing you with a pointer and with their fingers exactly what I'm showing you. So you want to investigate that also, okay? Just to hear it from somebody else. Okay, so now we're in the inner ear. Another name, bony labyrinth. Labyrinth. What is a labyrinth? I know what it is, but I'm not sure. Okay, no, you don't know. That's why you're not sure. A labyrinth is like a honeycomb, like a maze. And that's the way the inner ear is set up. The inner ear is set, I mean, take a look at it. It looks like a maze. Yes? Got it? There are three parts to the inner ear: semicircular canals, vestibule, and cochlea. Semicircular canals is for head orientation. They call it balance, but I don't want to say balance because it gets you confused with standing on one foot and the cat part of the brain, the cerebellum. Vestibule essentially is what I like to call the roller coaster. We get that feeling like our stomach comes up and stuff. It's not, it's in the vestibule. And the cochlea is for hearing. I think we got to cochlea in our prefix suffixes, which meant snail. Okay? So let's draw that sucker just to show you what it is here. Show you what it is here. What you see above my fingers, semicircular canals. You see the swollen part right here, as well as this part right here? Vestibule. You see the snail? Snail? Cochlea. So hearing, roller coaster, head orientation. And this represents the cranial nerve number 8, the vestibular cochlear nerve, used to be called auditory. And it's broken down into 2 parts. I'll get closer. Broken down into 2 parts. The cochlear and the vestibular nerve. The cochlear nerve, the vestibular nerve come together to form the vestibulocochlear cranial nerve number 8. Got that? Okay. I like to ask that on a lab exam. Okay. Let's draw what we've done so far. Okay. These are the semicircular canals. And there are 3. This portion is called the vestibule, roller coaster. This portion, cochlea, hearing. Okay. Semicircular. Let's do this one first. There are 3 of them. Anterior, posterior, lateral. Anterior, posterior, lateral. Lateral lays down. Anterior, posterior, lateral. Good? Okay. Now, I'm going to take a portion of one of those tubes. I'm going to take a portion of one of these tubes. I'm going to draw it. Okay, this portion right here is what it would look like right here. Do you see it right there? That is what I just drew there. And this is called a cupula. Cupula. Okay, inside of it, now this is a tube within a tube. So I'll draw the second tube in a different color. Okay, this is a cupula. And this part right here is called an ampulla. Now the other way around, never way around. I got that jacked up. I got that jacked up. Switch them. The outside is the ampulla, and the inside is the cupula. Do you understand? Now, think of the way we've been talking about the way the body's put together all semester. Why do you think that cupula is there? Look at it. Okay, before I answer, what I'm going to do is I'm going to tell you some of the components and see if you can figure this out. The outside fluid that lies between the black line and the blue line is called perilymph. It's thick, kind of like syrup. Inside is a thinner fluid, kind of like the way the eye was with vitreous humor and aqueous humor. And inside is endolymph, which is more like water. So what happens is both move, but one moves faster than the other. See if I can do this without screwing it up. Let's do it straight. Okay, that was jacked up. But anyway, now in green, I'll show you the last component before you tell me why that cupula is there. Lining this area is protein structures. Protein structures, kind of like cilia, but we give them a special name. We call them stereocilia. Okay? Stereocilia are connected to neurons. Okay, so essentially the stereocilia are protein that are connected to the dendrites. Get it? That's collected to the, that merges with the vestibular portion of the cranial nerve, vestibular cochlear nerve. So that's where it's coming out right there. We got it. See if I can do it a third time. Got it. Okay. What's the function of the cupula? I think you might have got it. It's called, I could hear you. It's called increasing surface area. So the function of the cupula is to increase surface area so you have more things moving, depolarizing. Does that make sense? Okay, so, and the outside portion of it, so it makes that bulb, is called the ampulla. That's the outside portion. Are we good? So let's go over that again. Semicircular canals, there are 3 of them. Anterior, posterior, and lateral. And they're not at 90-degree angles, like this. Why? No matter how you move your head, the endo length is going to pass over, it's going to move. If it moves, it's going to take stereocilia and bend them. When they bend, they get to a threshold of 55, they depolarize, and sends an impulse to the brain. You have learned as my head's to the right, my head's to the left, or whatever it may be. Do you understand? That's why there's 3 of them. You can't move your head without that fluid moving. Okay? And if you hold your head very straight without moving it, the fluid isn't moving. As soon as you go like that, the fluid moves, and you said you moved your head. Perilymph is the thick. Endolymph, thin. They slide across each other. Little cilia projections are called stereocilia, made out of proteins. To increase the surface area on the inside, you put them on a cupula. Looks kind of like an upside down U. The outside portion of it that contains it is called the ampulla. Wow. How'd you do? Okay. Let's go to vestibule. Okay, the second part. This is the part we're talking about right here. Now, let me erase that and do this again. It's set up very, very similar, but it's in a small area. So we saw this, the semicircular canals. In the semicircular canals, we had a large area to draw from. Okay, so that fluid can move in a long tube. You guys steal my marker? Guess I put it right here. Okay, I'm going to draw it again. This is the area we're going to be talking about right here. And there's a void, a lobule, a empty area. There's two of them. They're like a sphere inside of there. And one's called a utricle, and the other one is called a saccule. Utricle is for forward, backward detection. Saccule is for up and down detection. Now I'm going to take one of those, because they're set up the same way, and I'm going to draw it. Circle. Located in the circle are stereocilia. Okay. Inside, endolymph, outside perilymph, but the dendrites are not connected to the stereocilia. These are stereocilia. And what you have got dirt on my pink shirt. Are thicker and taller stereocilia. Thicker and taller stereocilia. And they have a name called kinocilium. Here's where I'm at. We're talking about saccule, utricle, stereocilia, kinocilium. Attached to the kinocilium is the neuron. You understand? So when the kinocilium bend, it causes depolarization, -70 to -55 threshold, then it depolarizes, sending electricity down here, which you know is sodium. Okay? Causes a depolarization. The stereocilia help push it. But you're saying, don't seem like enough. And if that did it, then when I felt the forward, backward, up, down, it would be very gradual. So what we're going to do is we're going to put chunks like rocks in here. Only they're not rocks, but they're similar. They're made of calcium carbonate. They're like little rocks floating in there. So when you move, those rocks help strike the kinocilium, and you get that reaction right away. You feel that right away. So what you're feeling when you go forward, backward, up, down, that dramatic movement of the body, the head, you're feeling the calcium carbonate that's suspended in the endolymph striking the kinocilium. The stereocilia also help bend that. But only the kinocilium is attached to the dendrite, the neuron. That organ, these organs, 1 and 2, collectively are called the otolith organs. Otolith organs. So the otolith organs are the saccule and the utricle. Got it? You know that it's made of endolymph and perilymph. Now you know what calcium carbonate is written up here before. And now you see vestibular nerve. Because remember, the vestibular nerve is this portion right here. This is the cochlear nerve. Together they make cranial nerve number 8, vestibulo-cochlear nerve. That's why I put vestibular nerve and not vestibular cochlear nerve, because we haven't got that far yet. Next thing you see, before I stop this for a minute, is the cochlea, which is the third part. So the third part begins with the oval window, which I just showed you. And I'm going to erase the board. Take a good look at it. I'm going to erase the board, come back, and I'm going to talk about how you hear. Okay? Okay, we're back. We've talked about the top portion of the inner ear, the semicircular canals. And you know that they're embedded with stereocilia, which are protein little hairs that stick up inside of the endolymph. On the outside of it is perilymph. The peri is thicker, the endo is thinner, so they both move. When the stereocilia bend, they're attached to a neuron, causes depolarization and the sense of movement. In between there is between my fingers now, is the vestibule. The vestibule is what we call the roller coaster. It's for forward, backward, up and down movement. It's called the otolith organ, and it's comprised of two spheres full of fluid. The spheres are the utricle and the saccule. Utricle is for forward, backward sensation. Saccule is for up and down sensation. Inside that sphere is stereocilia, but those aren't the ones that causes depolarization. It's the taller, thicker ones called kinocilium, and they're attached to a neuron. In addition, that movement that we get that we know right away that it's happening is because calcium carbonate rocks, crystals, strike the kinocilium as well as the stereocilia, bending it. The last one we're going to look at is the actual hearing, which is the cochlea. Okay? So here we go. Cochlea is for hearing. It begins with the oval window. So I'm going to draw the inner ear again. I think we also call that bony labyrinth. Okay, and remember that the bottom of the semicircular canal is the ampulla, and inside is the cupula, sacral, utricle, otolith organs, and now the cochlea. Right here, well, not right there. Right here is the oval window. Attached to the oval window, which I showed you before, was the smallest bone in the body called the stapes. So here's where I'm at. There's the cochlea. There's the oval window underneath the stapes. Now, what I'm going to do is I'm going to take this snail-looking thing and I'm going to unroll it. And when I unroll it, you're going to get a tube. And inside those tubes are 3 chambers. Okay? So I've unrolled it. Right here is a thinned out spot. I'll do it in a different color. There's your 3 chambers. 1, 2, 3. Now what I'm going to do is, this is where the stapes is attached. It articulates with the angle, the incus, which articulates with the malleus or malleus, the hammer, which articulates with the tympanic membrane. Okay, and this is your outer ear down here. Okay, now, in this tube and this tube are perilymph. In this tube is endolymph. And I hope you're getting it figured out by now. You got to have both fluids, so they're both moving, but the endolymph moves quicker than the perilymph. Okay? So now what I'm going to do is, I'm going to take a chunk of this out, turn it sideways for you. So here's the outside. This chamber and this chamber have a name. This is called scala, and this is called scala. Here's where I'm at. This scala is called scala vestibuli, and this one is called scala tympani. So now I got to see if the camera's catching that over there. Hang tight, hang tight. Barely. We're gonna have to move it over a little bit. Okay. Try to make it, when I made it big enough so you could see the words, it cut the board down too much. So, kind of sucks. Anyway, scala vestibuli is full of perilymph. Scala tympani is full of perilymph. And all those are is chambers to allow fluid to move by, the thicker fluid. In here is the third chamber called scala, has a couple names. Scala media, here's where I'm at. Also called the cochlear duct. Also called, and I'll put it right here because it's the big one, spiral organ of Corti. Okay, let's do a little check off. Talk about where is perilymph. Scala vestibuli, which is on the top. Remember, we unrolled this thing. We found out there's 3 chambers in it. Chamber number 1, chamber number 3, and in between is chamber number 2 that has a couple of names called scala media, also cochlear duct, also spiral organ of Corti. Now, the duct itself is called the cochlear duct scala media. That's the tube, which we're going to have stuff in there that is going to allow for hearing, and that is where we pick up the spiral organ of Corti. Got it? The fluid inside here is endolymph. Okay. Okay. I want to draw the scala media cochlear duct with all the stuff in it that makes up the spiral organ of Corti. And I'll do it right here. Kind of looks like this. And you have a lower membrane. And this is called the basilar membrane. Just like anything else made out of cells. On these cells, and I'll do it in red, some of those cells are attached to a neuron. And they have stereocilia. Remember what that was? Only I'm going to draw it a little bit bigger. Stereocilia. Now, obviously the stereocilia are going to move, and they're attached to axons that are collectively come together to form a cranial nerve, okay? So this cranial nerve that is being formed is called the cochlear cranial nerve, which is the other one that I've been showing you. Okay, now with the endolymph fluid, I've got to get my colors here. With the endolymph fluid moving back and forth, it's going to bend those stereocilia. But we don't want one bending and another one not. So what we want to do is we kind of want them all moving or not moving in unison. So what we're going to do is we're going to put another membrane on top of it to embed those stereocilia in. And that membrane is called the tectoral membrane. So watch, tectoral membrane on the top, basilar membrane on the bottom. Tectoral membrane is nothing more than just like a plate with the stereocilia embedded from the top. So this would be the tectoral membrane, this would be the basilar membrane, and the cells and the cilia sticking up into here. And you can see there's more down here because this is where the neurons are attached. These are just support cells. So let me go here. We have basilar membrane, support cells, dendrites, that's the red, or the cilia that's attached to the red. Tectoral membrane at the top. Stereocilia is what is going to move and is attached to the neuron. Okay, now let's kind of, how do I remember all this? If you go back to these senses, they all work the same way. You have dendrites that are initiating or being initiated to depolarize. So you have an impulse. It depolarizes, and collectively we take all of those axons, put them together and form a cranial nerve. We found that we're always using two fluids here, an endolymph and on the outside a perilymph. We also said that stereocilia, which are protein, small protein structures, very much like cilia, only it's dealing with the inner ear, so they put stereo in front of it, is going to be moving and attached to a neuron, except in the vestibular gland. I mean, the otolith organs, sorry, in the otolith organs. And what happens is you move them and the stereocilia move. When the stereocilia move, it causes depolarization, -70 to -55 threshold, and then away it goes. We know that there's three parts to the inner ear. The inner ear is where everything happens. The outer ear, the middle ear, is just to get that message into here. If we move our head, we're doing two things: the semicircular canals and the otolith organs, the saccule and the utricle. If it's hearing, what it's doing is it's causing the stapes to move in and move out. Oh, hell, I didn't do that for you. Okay, we made stereocilia red. So these are all made up. What did I do? I made it out of blue, just so I'm consistent. These are cells. I'm drawing the walls of the cell, or the phospholipid bilayer of the cell. Okay, and we said every now and then we have a cell that's attached to a neuron. And extending up from there is stereocilia. And then there's a tectoral braid. What do we do? We did it in green. There's a tectoral membrane. You see where I'm going with this? So what happens is when the ossicles push in, it causes endolymph. What color did I make endolymph? Green. It causes endolymph, I'll put endo, to move. Okay, so it moves this way. When it pulls back, it moves the other way. So it's moving the stereocilia like this. The more that are involved, the higher the pitch, or the louder it is. Okay, the pitch. The frequency is where it strikes on here. So lower frequency is going to go all the way through. Higher frequency is going to be short. Do you understand? Okay. A in front of any word means without. When you see the, remember we see opsia meant eyes, aphasia means speaking. Inability to speak, aphasia. Meissners disease. Tinnitus, ringing in the ears, Tinnitus. Tinnitus, the ringing of the ears. When it doesn't go away, it's called Meissners disease. That can also lead to vertigo, you know, where your feels like your head's spinning and you've had too much to drink, trying to listen to one of my lectures. Okay, let's see where we're going from here. I'm not going to finish up today. I'm not going to finish up the cardiac. So I'll come in Friday and do the cardiac. It'll be set up just like this. 3 steps: cardiac, the heart, blood vessels, and blood. That will take you in to way past next week, okay? Up to the exam. Let me caution you on a few things. Sitting at home in your pajamas with your headphones on and only paying half attention, you're going to dump the semester. You got to stay focused. If you can't find a place to go, and we're supposed to have that kind of isolation, is it? Anyway, we're supposed to be 6 feet away from somebody. That I totally understand. And running to the library that's closed isn't going to work. You may have to go out into your car. You've got Wi-Fi, you've got your cell phone. Don't give me that kind of stuff. You know you have it. So focus on that. Listen to this. Stop it when you don't hear something, or you think you hear something wrong, or I say it too quick. Stop it, go back, and do it again. If that doesn't work, and you still have questions, either go on Canvas and ask the question, or go straight to email or Bio31 on Facebook, and we'll get it answered for you. Okay? I know you may not like this. I don't like this. I don't think it's the best modality, but it's the only one that we can offer you right now. Okay? Social distancing. Okay? Practice that. We will get over this. Okay? The peak hasn't been reached, but we're suppressing that peak. So what we want to do is, this is the way any virus goes, okay? That the least amount of people that get it, or the people that just barely get it, build up an immunity and they're okay. And once we get back to that, we can take it from there. Okay? Any questions? First go to Canvas, then go to Facebook, then go to email. Are we good? All right, I'm going to shut it off. We'll get these uploaded today, which is Wednesday, so you'll have them to look at tomorrow or tonight. See ya.